Hello and welcome to Chapter 7. In Chapter 7 we're going to look at cell structure, and today we're going to look at Section 1, which pertains to the introduction to cells. In Section 1 we're going to look at how cells were discovered, why the cell shape varies, and what enables eukaryotes to perform the more specialized functions than prokaryotes. As you can imagine, the microscope observations of organisms are what led to the discovery of basic characteristics among all living things. It was somewhere in the 1600s that scientists were able to first discover the cells, and these were using really crude microscopes, which for the time, being in the 1600s, the microscopes, although they were not nearly what they are today, it's pretty impressive that they were able to still find something as small as a cell. And it wasn't until the 1800s, when microscopes had kind of been refined a little bit more, that observations made by German scientists were used, and these observations are what led to what we call the cell theory, or the formation of the cell theory. And the cell theory is actually, it's three parts to it, and it kind of lays the foundations, and these three parts are something that you will need to know. So the first part of the cell theory says all living things are made of one or more cells, okay? And that's everything. Anything that is living is made up of at least one or more cells. The second part of the cell theory says the cell is the basic unit of structure of living things. And finally, the third thing is cells arise from existing cells. So you have to have a cell to create another cell, essentially. So please take a quick moment, make sure that you have these all written down, and make sure you know and understand what these three things are, because as I mentioned, these are very important and you will be required to know what these are. So as one would imagine, when we look at cells, cells are going to vary greatly in size and shape. Okay, and ultimately that's because it's going to depend on what that cell's function is. So if you look at something like a skin cell versus like a hair cell versus like a lung cell, okay, all of these cells have different functionalities. Therefore, their size and shape is going to differ based upon what they're responsible for doing. So we can say that a cell's shape reflects its function, and the size is limited by the cell's surface area to volume ratio. And we're going to talk more about that here in a little bit. Cells can also be branched, flat, round, or rectangular. So their shape can vary, again, based on the function that they have to perform. And any substance that wants to enter or leave the cell has to cross the surface of that cell. And it seems like a pretty simple idea, but it's kind of a complex process to be able to cross that surface. Okay, and the cell's ability to move a substance across that surface can be estimated by finding its surface area to volume ratio. Cells that have a greater surface area to volume ratio can exchange substances more efficiently than those that have a smaller surface area to volume ratio. So how the surface area to volume ratio works is, if we just take something like simple like a cube, okay? Notice here we have three different cubes. Okay, the first cube we have is a 1 millimeter by 1 millimeter by 1 millimeter cube. Okay, so if the side length is 1 millimeter, our surface area is going to be 6 millimeters. And we remember, if you find surface area, I take the length times the width of a surface. Okay, so that in this case, because it's a cube, 1 times 1 is 1, but I have 6 different faces. I have 4 sides all the way around, and then I have a top and a bottom. So my surface area in this case is going to be 6 square millimeters. When I find the volume, I have length times width times height, or 1 times 1 times 1, which is going to give me 1 cubic millimeter. So if I do surface area to volume and I set this up as a ratio, I get 6 to 1. 6 square millimeters for my surface area, 1 cubic millimeter for my volume, and that becomes my ratio. Now when I look at my 2 millimeter block, okay, I'm 2 millimeters by 2 millimeters. So in this case, if I go to find the surface area, I have 2 times 2, which is 4, but then I have to multiply that by the 6 faces, so that gives me this 24 square millimeters. And to find the volume, I have 2 times 2 times 2, which is 8. So I end up with 24 to 8 and 24 to 8. So this gives me 24 to 8, which will reduce down to a 3 to 1 surface area to volume ratio. Likewise, when I look at my 4 millimeter ratio or cube, I have 4 by 4 by 4. I'm going to end up with a surface area of 96, and 4 times 4 times 4 is 64, so my volume is going to be 64 cubic millimeters. And when I do my ratio, this here would turn out to be 96 to 64, or if I reduce it, I end up with 3 to 2. So based on this information alone, you can see that your largest surface area to volume ratio is actually going to be the one with the smallest cube, or this right here. So this is going to kind of confirm the fact that when we compare cells of the same shape, the small cells are going to have a greater surface area to volume ratio than our larger cells, which might be kind of a little bit difficult to wrap your head around. Because typically we would think that the larger the cell, the larger the surface area to volume ratio, but it is in fact, based on the scenario we just walked through, it is in fact the opposite of that. So the smaller the cell, when we're looking at things that are the same shape, the smaller the cell, the higher the surface area to volume ratio. So ultimately what that tells us then is small cells are going to function more efficiently than larger cells will. When we're looking at different types of cells, they have some common features amongst them, okay? And some of those include the cell membrane. These are structural features. We have cytoplasm, we have ribosomes, and DNA. The cell membrane is that outer layer that covers the cell's surface, and it actually is the barrier between the outside environment and the inside of the cell. So it's kind of what keeps things, keeps the inside stuff of the cell on the inside, and it keeps the outside stuff on the outside. The cytoplasm is a region of the cell that's inside of the cell membrane, and it includes the fluid that we call, or inside of the cell, that we call the cytosol. So the cytoplasm is the region inside of the cell membrane, and the fluid is called the cytosol. So cells are typically going to have ribosomes, and a ribosome is a cellular structure that actually makes the proteins for the cell. And we're going to talk about the proteins and the importance of those later on. And finally, some common features when we talked about DNA. Remember that DNA is the genetic information of a cell that provides instructions for making proteins, it regulates the cellular activities, and it's what enables the cells to reproduce. And when we look at cells, okay, we're typically going to have two different types. We're going to have prokaryotic cells and eukaryotic cells. So just to kind of look at the differences, notice over here we have like these little hair-like things called pili. The DNA is on the inside. We have ribosomes, which are kind of like these little red dots floating around. This cell here actually has a cell wall. The membrane is underneath that cell wall. We have over here, we can see that we have a nucleus, we have a cytoskeleton, we have a cell membrane, just like on the other cell. We have something called smooth and rough ER, we have vesicles and mitochondrion. So this one over here has a lot more stuff than this one over here. And we're going to talk about the differences between the two and why they are what they are. So the first type of cell that we're going to talk about is what we call a prokaryotic cell. A prokaryote is an organism that's made of a single prokaryotic cell, and these were formed about 3.5 billion years ago. Prokaryotic cells do not have a nucleus, okay, and we didn't see a nucleus in the cell that was on the left here a few minutes ago, and they do not have other internal compartments. The genetic material is also just a single loop of DNA inside of that prokaryotic cell. Okay, and for the longest time, okay, like millions of years longest time, prokaryotes were the only organisms on Earth. The other type of cell that we're going to talk about is called a eukaryotic cell. And a eukaryote is an organism that's made up of one or more eukaryotic cells. So typically when we're looking at something that's multicellular, like we are, okay, we're made up of many eukaryotic cells. So in the prokaryotic cells, we talked about how there was just a single strand of DNA inside of the prokaryotic cell. When we're looking at the DNA of a eukaryotic cell, this is found inside of a compartment called the nucleus. Another difference is that eukaryotic cells have what we call membrane-bound organelles. And an organelle is just a small structure that's found inside of that cytoplasm, okay, and that it will carry out a very specific activity inside of the cell. And you're going to see we're going to talk about different types of organelles over the course of this unit and kind of what those organelles do. Each organelle is going to perform a very distinct function within that cell. And it's the complexity of the eukaryotic cell that enables them to carry out very specialized functions, which a prokaryotic cell can't do because it doesn't have all that, the complex system and stuff that a eukaryotic cell does. So just to do kind of like a side-by-side comparison here, when we look at the prokaryotic and the eukaryotic cells, a prokaryote does not have a nucleus, where a eukaryote does. A prokaryote has no organelles in it, to where a eukaryote is going to have many organelles. The size of a prokaryote is 1 to 10 micrometers, which is pretty tiny. Okay, and then you have, and for eukaryotic cells, we have 2 to 1,000 micrometers in size. The prokaryotic cell is about 3.5 billion years old, to where a eukaryotic cell has evolved over time, and these are only about 1.5 billion years old. And then prokaryotic cells are only bacterial cells, to where eukaryotic cells are all other cell types. So these are kind of like the key differences between the pro and the eukaryotes. And this is where we're going to stop for today. Please let me know if you have any questions. Otherwise, I will see you guys in class. Have a great day.